Didn't mean to unshare it. All right, did anyone look at any of these little clips that we've been making? Am I wasting my time or is it worthwhile? As I kind of record the class and put it up there. I think we'll have a better understanding when we get closer to a test. We'll have a better understanding. So let's just we'll keep doing it for now, and so forth. So again, quiz on Friday, right? And it's basically going to be some labeling and some organelles. Nothing too crazy. Mitochondria, right? We talked about this last class, correct? We talked about mitochondria, we talked about chloroplast just briefly. All right, so just give a little bit of detail. We don't need a ton of mitochondria, right? Because Andrew, it is definitely what? The powerhouse of the cell. Very good. Just a couple things with the mitochondria that we should be familiar with. It has one membrane. It's a double membrane. It has a double membrane, which is important to its function, which we'll talk about later. Also, the inner membrane has all these folds known as, starts with a C, cristae, all these extra folds, cristae. And what do these folds do? It increases the effectiveness, it increases the efficiency of a mitochondria, allowing it to convert more energy from glucose into ATP, but it also does what? How does it do that? By increasing what? Increases surface area. Very good. That's what it does. It's a powerhouse of the cell. So what that means is it takes the $10,000 bill like a bank and breaks down into 5s and 10s and 20s to use. Takes the big energy glucose and breaks down into ATP, usable energy. Okay, couple things about mitochondria. They have ribosomes. Why would mitochondria have their own ribosomes? On the same question, they have their own DNA. Why do mitochondria have their own DNA? This is where I, kind of things I would take notes, or these are things that are not in a PowerPoint. Okay. Mitochondria have their own DNA, they have their own ribosomes. Even stranger, mitochondria replicate on their own. When your cell divides, it does not make new mitochondria. The mitochondria divide on their own. What is this telling us about a mitochondria? What's that? They do not have a nucleus, but you're not in the right ballpark a little bit. They evolved, right? Mitochondria, their ancestors were their own individual prokaryotic cells. So mitochondria, way back when, billions of years ago, mitochondria were actually just a single-cell prokaryote. And it became a part of a eukaryotic cell. I'll give you the term. I'm going to mention this again kind of when we talk about evolution, natural selection, kind of the tree of life. So you don't need to know this for this test. But it's known as endosymbiotic theory. So it's how the mitochondria started to work with the bigger cell to go from prokaryote cells to eukaryote cells. Yep. I will not push you directly into symbiotic theory, but since we're talking a little bit about mitochondria, we can mention it. Guess what? Chloroplasts have ribosomes in their own DNA as well. What does that tell you about chloroplasts? They were once their own prokaryotic cell. Endosymbiotic theory didn't happen once, it happened twice. Once when the mitochondria came in, and another time when chloroplasts came in. What do chloroplasts do? Absolutely. Chloroplast is where photosynthesis takes place. What is photosynthesis? What's that? Very good. It converts sunlight energy into food energy, right? I like how you said that, because a lot of people just say it makes food, which is implying that it makes energy. It does not make energy. It simply converts energy from one type, light energy, into chemical energy, food, sugars. So our chloroplast is very simple. Right now, it's where photosynthesis takes place, right? And it's where light energy gets converted into food. What group of food molecules should we focus on? What type of molecules do they make? What's that? Who said it? What's that? Glucose. Very good. It makes glucose, carbohydrates. I know you're so upset that you're not going to have biology class. I'm sure. I'm sure. All right. There are some key structures with chloroplast. I'm not too worried about them right now. We'll deal with them when we talk about photosynthesis. But thylakoids are the little discs there. Granums are the stacks, and the stroma is the space in between. Just so I mentioned the term. But right now, you don't need to know that. Just know chloroplast is where photosynthesis occurs, converts light energy into food energy, glucose, carbohydrates. And the one kind of last one that's really part of the metabolic process are peroxisomes. So here's a chloroplast over here. Here's a mitochondria over here. Here's a peroxisome. And all it does is this. The processes of photosynthesis and the processes of cellular respiration produce some pretty nasty byproducts. Right? Oxygen can do damage if it's not dealt with. Right? Oxygen can break down other molecules. So the peroxisome is there to kind of deal with the byproducts, deal with the oxygens. And it forms hydrogen peroxide, H2O2. So not really a huge thing, but they do serve a function, and basically they're kind of like a cleanup system for the mitochondria in the chloroplast. It turns it into hydrogen peroxide, and it's peroxide as well. Give me a second. Peroxide, I mean, just go with basic stuff. It's not big. Okay, so now we're going to talk a little bit about the cytoskeleton. And then the reason why we group, actually, you can tell me, why do I have the cytoskeleton grouped with centrioles, cilia, and flagella? Cytoskeleton are on the inside of the cell. Cilia and flagella are found on the outside of the cell. Centrioles are involved with what? Cell division, starts with an M. Mitosis. So why are they all grouped together on the slide? It seems like a little bit of an odd grouping. They serve completely different functions, but they all have similar features, structures. Very good. They all have similar structures. So like when we talk about the cytoskeleton, we talk about microtubules, microfilaments, intermediate filaments. Those structures help to make up our cilia flagella as well. Microtubules, more specifically. So they're grouped together because they're kind of made of similar structures, the microtubules. Okay, but they have very, very different functions. So let's deal with the cytoskeleton first. Is the cytoskeleton a dynamic or a static structure? The cytoskeleton of the cell, picture, right? All that network inside the cell. Is it a static or dynamic structure? It's a dynamic structure. It moves. It shapes the shell. It's a transport system. So it is not static. It is dynamic. The cytoskeleton is comprised of three main components: microtubules, microfilaments, and intermediate filaments. Two of the three are dynamic, are involved with movement. One of them is a static, really just kind of a connection structure. Which one of the three is really just used for connections? Static. The intermediate filaments. They're more just kind of anchoring pieces. They don't really move too much, not involved with movement. The microtubules and microfilaments are involved more with movement. So when we look at microtubules, we have some key functions. You do not need to memorize all of this. Don't worry about this. You don't need to go this specific, okay? What do you need to know about microtubules? They help with cell shape and movement. That's it. Microtubules help with cell shape and the movement. They're the largest of the three. We should kind of note they're the largest of the three. But cell shape and movement. So microtubules, cell shape and movement. And movement. Couple things. You remember the video? Remember how the video had the train tracks were moving things from one to the other, the vesicles being moved around? Well, what they're moving on are microtubules. So that's why they're kind of like the train tracks themselves. But microtubules also can help make up cilia flagella and help move things around that way. Everyone okay? All right. Microfilaments helps maintain cell shape and allows for cell movement. They both have similar functions. You don't really need to differentiate between the two. So maintain cell shape and helps with movement. Same thing. It's just like tubules. Again, you don't need to go too specific between the differences between the two. There are some differences. They're not exactly the same, but for our purposes, we don't need to worry too much about that. Everyone, a couple things. An amoeba. Everyone knows an amoeba, right? You've heard the word amoeba before. It's a single-cell protist that moves through a movement known as amoeboid movement. So what happens is I have this amoeba, which can dramatically change the shape of itself. And what happens is the amoeba can stretch out part of its arm, actually stretch its cytoplasm out, and reach out and grab on, and then slide its body over. So you can kind of picture like a blob moving. That's how an amoeba can move. Now, how does it have the ability to stretch its cell membrane out? The microfilaments, right? They're inside. They can stretch. They can pull. Everyone picture a cell dividing, right? So I have a cell where mitosis has occurred. So eventually that cell, if it's an animal cell, that cell starts to kind of pinch off into two cells. What's controlling the pinching of the cell membrane? Bless you. Microfilaments, right? They're pinching the cell membrane together. So that's what we talk about with movement there. The last one are intermediate filaments, and these are really just kind of connection places. So they're more rigid. They're just kind of there to maintain shape, but they're also anchorages. Your skin cells aren't just like hanging out next to each other. They're actually connected to one another, right? And how are they connected to one another? Some of these intermediate filaments kind of form these, they're known as desmosomes, but kind of make these connections between. So this is more for connection, and these are rigid. Everyone okay? Oh. We're good? Okay. So here's just kind of a picture of a little microtubule. You see this whole meshwork here. The thinner ones are the microfilaments, the larger ones are the microtubules. Again, they form that framework of our cytoskeleton. So the one thing I just want to kind of talk about, and we will finish up with this, is just this idea of motor proteins. So proteins serve a function of they're enzymes, but proteins also serve a function of movements. And the question is, so we have these microtubules, which are the tracks. Well, how do vesicles actually move down the tracks? So I kind of liken it to a monorail system. Have you ever been to Disney World? I'd like to go again. I haven't been in a long time. So it works like a monorail system, right? Where you have kind of like a train on a single track moving along. It's similar to a train. This is just one track. But this is how it works. Here's my little protein right here. And here's the vesicle I need to move. The vesicle doesn't just slide down a track and go. The vesicle's not designed to move. But this motor protein is designed to not only hold on to the vesicle, but it's designed to move. Okay. Here's what happens. Now remember we talked about, the currency of energy in our system is ATP, right? And what ATP does is it energizes molecules. In this case, it's going to energize a motor protein. Now, when it energizes a motor protein, that's kind of vague language. What do you mean by energize the motor protein? Well, what happens is that ATP, remember ATP can kind of give up a phosphate group. ATP goes to ADP. Remember that at the very end of nucleic acids? So what happens is a little piece of ATP gets transferred from here to the protein. And whenever you add something to a protein, that protein kind of changes its shape. It's that simple. So when ATP transfers the phosphate group, if I was the motor protein, right, when the phosphate group gets added to me, so let's say this nice little magnet is ATP. This represents the phosphate group. When the phosphate group is transferred to me, it causes me to do this. Move one piece forward. Because this phosphate group causes me to change my shape. When I give it up, it causes me to go back to my initial shape. Another ATP comes in. Gives me another phosphate group. Causes me to change my shape. And I go like this. I get that phosphate group back up, and I go back to my original shape. So you can see this motor protein right here is attached to the microtubule. Whenever it's energized, one of the legs moves forward. Whenever you take that phosphate group off, whenever it's de-energized, the back leg catches up, goes back to its original square shape. Square, uniform shape. You energize it again, it moves forward. You take it off, it goes back. Right? And simply, that's how these motor proteins work. So in that video, we saw these little vesicles moving all over the place. Well, what's actually doing the movement are these little proteins, all these little walking proteins. Pretty straightforward, pretty simple. So they're energized, they take a step forward. They're de-energized, they catch up. Energized, take a step forward. De-energized, they catch up. And you just keep doing that over and over and over and over and over again. And that's kind of how our motor proteins work. All right, we will finish there.